Welcome everybody for our weekly seminar series. Today I am very honored to present you, whoever don't know yet, Dr. Laura Streib, who is a postdoc affiliated to both University of Kentucky and University of Haifa. And Laura is a Zuckerman postdoctoral scholar, as I say here in Haifa and at the University of Kentucky. Her research uses lake sediments to study past climate and environments. Her work as part of the Zuckerman program centers on Lake Tanganyika in Eastern Africa. Lake Tanganyika is the world's longest and second deepest lake. This project seeks to understand the human influence on Lake Tanganyika over the past 2,000 years and has the ultimate goal of determining whether natural environmental perturbations or human activities control the lake fishery. As a PhD student at Syracuse University, she studied 1.4 million years old drill core from Lake Malawi, also in Eastern Africa. This record was collected in 2005 and represents the longest and most continuous sediment archive from the continental tropics. But it was unfortunately underutilized for many years due to chronology uncertainty. Dr. Streib work refined the chronology for this record using luminescence dating and generated a new hydroclimatic story using Diatom's Paleoecology. She also has an ongoing work on a 1.1 million years old drill core from Lake Bosumtwi in Western Africa. Like the record from Lake Malawi, this core has been underutilized since its collection 20 years ago, but can now be reanalyzed using updated methods. So today we are going to hear about Lake Tanganyika, and the podium is yours. Thank you very much.
Okay, great. So my name is Laura Streib. I'm a postdoc in Nicholas Waldman's lab. My research uses lake sediments to study past climate, and right now I mostly work on lakes in Africa. So I'm going to tell you a bit. I need the mouse, or maybe.
No. Go ahead.
There we go. So my research mostly focuses on Africa, and I'm going to tell you a little bit about 3 different projects on 3 different sites today. Can you close so that we don't see the?
Yeah.
So 3 lakes. We have Lake Malawi. That's a 1.1 million year record over here in Eastern Africa. This part is going to be on chronology and hydroclimate. A record from Lake Bosumtwi, way in West Africa. That's a 1.1 million year record. It's going to be about chronology. And I'll tell you just a little bit about the current project that I'm at Haifa to do. That's a 2,000 year record for Lake Tanganyika. It's about nutrient cycling. So these projects on Lake Malawi and Lake Bosumtwi are quite similar. These are both archived drill cores that were collected more than 20 years ago. But they had serious problems with chronological uncertainty, and they weren't really used to their full potential. So what my work did was go back and redate these records using luminescence dating, so that we can then reanalyze what they tell us about hydroclimate and what's driving our system. So I'll start with Malawi. That's what I'll spend the most time on. Right. So first, Lake Malawi chronology. So Lake Malawi, at 550 kilometers long, 700 meters deep, and more than 5 million years old, it's one of the world's largest and oldest lakes. Because our lake is so large, climatic perturbations are muted, and regional climatic change is recorded. And because our lake is so deep, it never completely desiccated during the Quaternary. Oftentimes when you're studying lake records, small lakes will dry completely during the record, and you'll end up with gaps. Because our lake is so deep, this isn't a problem. We have a record of a complete continuous record. Right, so why does this matter? Lake Malawi has more fish species than any other lake in the world, many of which are endemic cichlids. This makes it a good place to study relationships between evolution and climatic changes. We need to have a good sense of the timing of these climatic changes to make those connections. Because humans were evolving and developing in Eastern Africa, they were in some way shaped by the climate in the region. And today, communities in Eastern Africa are experiencing explosive population growth and extreme climatic stress. If we want to both protect these communities and understand the relationships between past organisms and their environments, we need to understand the climatic system in Eastern Africa. So the drill core that I'm working on, it was collected in 2005 from 600 meters of water depth. It's 380 meters long and goes back to 1.4 million years. It represents the longest, most continuous sediment core from the continental tropics. But like I said, it has a gap in its chronology that limited its use. So what my work did was use luminescence dating to try and fix this problem. So luminescence dating for sediment core, the people that work on the continents, for quite a while, there was a big problem of dating in between radiocarbon dating that goes up to around 50,000 years. And then you have this big gap before you get to paleomagnetic excursions that start around 800,000 years. And unless you have things like tephras or carbonates in your core, there are few options for dating. But luminescence dating, which has been developed much more recently, it can work on quartz and feldspar grains, with quartz being for younger samples and feldspar being for older samples, has more recently been able to fill most of this gap. Right? So when these cores were first collected, there weren't a lot of options for dating in this sort of 50 to 800,000 age range. And with luminescence dating, now we can go back and fill that. I have a picture of a dune here. Because luminescence dating was historically in these driving systems, and only in the past few years have we started applying it to lakes and other environments. Right. So what we're looking at here is a stratigraphic column for our core with the previously existing dates. And you can see here that gap highlighted in red from 74 to 590,000 years. Right, that we're trying to fill. So for our record, there were 3 published age models. They either didn't fill this gap in core, they tuned a temperature record to the marine benthic stack, or they used paleomagnetic excursion and intensity dates. So each of those age models, we have here depth on our core on our x-axis and age on our y-axis, are each of these 3 lines. And you can see that depending on the method that they used, people got really different answers about what the age was for this core. Because they got these different answers in chronology, they ended up with different answers about what was driving the system, and it became a problem. Right? So what we're doing is trying to fix this problem with luminescence dating. Right. So a bit on the methods that we used here. We've got back to our strat column, and all those yellow triangles are all of our new ages. For part of this, we did a blind trial to see that our ages were matching the previously published radiocarbon ages, a more trusted method. We got our samples reflected at a 4-meter resolution throughout our interval of interest. All this work was done at Royal Holloway, University of London. I went to that university for a semester to do this, and we did it all on the 4 to 11 micron size fraction and followed a single aliquot regenerative dose procedure. If you don't know what that means, that's pretty much just the standard procedure for luminescence ages. So I want to note too that calculating water content ended up being a really important part of this project. Water content is a necessary part of luminescence ages, with a 1% change in water content gives you a 1% change in age. Unfortunately, because our core was collected back 20 years ago, people either didn't collect or didn't preserve the water content data. So a big part of this project ended up being figuring out a way to calculate water content with the data that we did have, and then calculate dewatering. Often with luminescence dates, they're getting near surface ages, but what we had was sediment that was buried and then had been progressively compressed over the last few 100,000 years. So we did not only calculate the water content of our sample, but figure out how water content had changed through time. And that ended up being quite important to our eventual ages. On the other thing, this is our blind trial results. So we have in blue here, radiocarbon ages, and our new dates are those yellow luminescence ones. They agree quite nicely. Because luminescence dating is sort of new to the discipline, it was important to show that these agreed. And moving on to our new chronology. This is our age model. We again have depth on our x-axis, age on our y. All those new yellow dots are our new ages. And hopefully you can see that we were able to fill that gap with our absolute ages now. And what we can do now that we have a chronology, they start to move forward. Right? So our chronology contributes to work showing that luminescence dating is now well suited for Lacustrine records, and we can apply it to other records like this one. This project also contributed to work that shows that water content history can be effectively calculated from sediment cores that have been stored for long periods. And our new age model fills this gap, allowing this gap in chronology, allowing for a reassessment of the hydroclimatic record and more high resolution work. That's what the next section of my talk is going to be, the reassessment of our hydroclimate record based on our new chronology. All right. So moving on to that, the Lake Malawi hydroclimate record. Okay, so for this part of the talk, I'm going to be talking about two different kinds of proxies: diatoms and potassium titanium based on XRF scanning. So our diatom record extends to 420,000 years at a 450-year sampling resolution. And our potassium titanium record extends for the full length of our core at a 50-year sampling resolution. This is based on core scanning, so it can be a much higher resolution because it's a much less labor-intensive proxy. Okay, so I'll tell you a bit about how each of those proxies work. So diatoms are single-celled algae, and they have skeletons made out of silica that preserve well in sediment. There are planktic diatoms that live in the water column, and their abundance and diversity is largely driven by changes in nutrient status. So we're talking about planktonic diatoms today. Oftentimes in lacustrine systems, nutrient status and therefore diatom assemblages are controlled by the strength of mixing, right? That's how water is moving through our lake, which is often linked to seasonality. But at Lake Malawi, nutrients, and especially silica, can be permanently buried and have to be replenished from the landscape. Because our lake is so deep, once that silica gets down to the bottom, it can't get back up into our water column. So at Lake Malawi, the diatom assemblage is driven by rates of precipitation, where when we have lots of precipitation, we have lots of silica driven into our lake, and we're going to get blooms of diatoms that require lots of silica, like this one. And during periods of much lower precipitation, we're going to get blooms of these lower silica diatoms. Our high silica diatoms have much thicker skeletons. So sort of by necessity, they require higher silica. And you can see this one here has a much thinner skeleton. It's going to require less silica. Okay. So we can take a ratio of those two kinds of diatoms as a hydroclimatic indicator. So the other proxy we're using is a ratio of potassium to titanium based on scanning XRF. So this proxy works by precipitation influencing vegetation, weathering, diagenesis, and the rate of lake outflow. So the way this works is that during wet intervals, we have an environment with lots of vegetation, so our soils are stable, and there's lots of chemical weathering that's going to make this potassium soluble. What that means is that most of our sediment is going to stay on our landscape, and not a lot of it is going to go into our lake. And sediment that contains potassium is going to flow out of our lake via riverine discharge. But when we reverse this during dry conditions, we're going to lose all that vegetation. It means our soil is less stable. We're also going to decrease chemical weathering. That's going to mean that a whole lot more potassium is going to go into our lake. And the potassium that does enter our lake is going to stay at our drill site rather than flowing out of our lake via an outflow, right? Once our lake is low enough, we're going to lose that outflow, right? That often happens with lakes. When you have a high lake, you have riverine discharge. Once you get below a certain level, you're below that riverine sort of maximum, and all the sediments stay in your lake. So what this means is that we can look at potassium as a dry indicator. During periods when we have more potassium, we're dry. During periods when we have less potassium, we're wet. And then we use titanium, which isn't influenced by any of these processes, to sort of normalize our data set. Okay. So on to interpreting our data. We have our two data sets here. We're zooming in here on the length of our diatom record, a diatom record up top, and our potassium titanium record down below. They generally agree with each other. We're looking here as dry indicators facing up. And what we can also do, there's a whole lot of wiggles here. What I'm showing you, these two are our proxies, and these are other proxies available from the core. There are also hydroclimatic indicators. And what we want to see is that generally our proxies agree with each other. They're not in perfect alignment. Each proxy responds to the hydroclimatic cycle differently, and the ways they disagree make sense for what we know about them. But in general, we can use these different proxies to confirm some of our interpretations. All right, so moving on to what this tells us about what's driving our system. So first, look over here at this figure. We have our diatom record, our potassium titanium record again, and I've plotted this up against a sea surface temperature record from the Arabian Sea. Hopefully you can see here. So our data is oriented dry to the right, and this data here is oriented warmer to the right and cooler to the left. And hopefully when you look at this, you can see that warm sea surface temperatures tend to be associated with dry conditions, and cool sea surface temperatures are associated with wet conditions. And this is really important because it runs sort of exactly opposite to a lot of previously held assumptions about climate in Eastern Africa. So we've zoomed in here on our diatom record, but this relationship generally extends for the length of our. All right. So an important part about this observation is that it effectively requires that Lake Malawi is behaving opposite to sites to the north and south. So we're looking here at our Lake Malawi record in the middle, oriented dry up, and these other records to the north and south, oriented dry up. Help you sort of see that relationship. They don't align exactly, but if we're to say nudge them a little bit, this is all, we've moved them around a little bit to just to show that alignment better. But the amount that I've moved them around is all within the error of our chronology. So they're all possible ages, the things that could have happened. I hope we can start to see that they come into alignment. Or when Lake Malawi is dry, these other sites are wet, and vice versa. It can be hard to see with all these wiggles, but we can confirm this with age uncertain correlations and that sort of thing, different statistical analyses. Okay. So the remaining question is, why would we see this relationship? Why would Lake Malawi behave differently from all these other sites? So precipitation in our region today is driven by the north-south migration of the tropical rain belt. That's that sort of band of purple in this figure here. So to create this pattern of anti-phase hydroclimate, where Malawi behaves differently from these other sites, we need to alter the path of that tropical rain belt. The path of the tropical rain belt is controlled by the north-south energy gradient, drifting towards the warmer hemisphere on geologic timescales. Okay? And another important thing is that hydroclimate in southern Africa, where these two most southern sites are a little bit different, and they tend to respond directly to changes in sea surface temperature. So I'm going to take you through how this process works. So as a general outline, so what we think is happening here is that our system is driven by changes in ocean circulation, induced by glacial and interglacial cycles that alter the north-south temperature gradient in the Indian Ocean, which in turn influences the path of the tropical rain belt. So that's a lot, but we're talking about how different currents are going to change. That's going to impact temperature in our Indian Ocean. And as you change temperature in your Indian Ocean, you're going to change the pattern of precipitation. So we'll walk through this. We start at interglacials. Interglacials are our warm periods. During our warm periods, Malawi is dry, and these other sites are wet. And the first component of this over here is called our Indonesian through flow. It's a really important current for global circulation and global temperature, and it changes through time or linked to a lot of different things. Based on a modeling paper from 2023, sort of paradoxically, during warmer periods, our interglacials, we have what are called freshwater lenses forming over here in the South China Sea. And what those freshwater lenses do is they prevent warm surface waters from flowing through this current, and they make this current much colder than it would be otherwise. So what happens is we have this cold current overflowing out of Indonesia, and this cold current is going to cool this south equatorial current here. And what that's going to do is cool our southern Indian Ocean relative to our northern Indian Ocean. And if you remember a minute ago, I said that our tropical rain belt is often linked to changes in temperature gradient, drifting towards the warmer hemisphere. So what we've done is made our southern Indian Ocean colder relative to our northern Indian Ocean. And that's going to cause our rain belt to drift north. And it's going to cause these northern sites to be wetter and Lake Malawi to dry. At the same time that that's happening, our subtropical front down here is going to be much colder. And our subtropical front is a current that circles Antarctica and it moves with glacials. Because we're in an interglacial, a warm period, it's way down south. And we have lots of warm water off southern Africa. And that's going to make those sites wet. So during our interglacials, we have a cool southern Indian Ocean and a relatively warm northern Indian Ocean, making our northern sites wet and Lake Malawi dry. Well, at the same time, this southern current is going to stay nice down in the south and keep these sites wet. Then during our glacials, our cold intervals, we're going to reverse this whole thing. During our cold intervals, Malawi is wet and all these other sites are dry. Right. So during these intervals, the first thing to note here is that during a glacial, lots and lots of ocean water is locked up in glaciers. So we have much reduced sea level and a lot of land exposed, right? So now because we have all that land exposed, those freshwater lenses that were stopping warm water from flowing through here, they don't exist anymore. And now we can have all this warm water flow through that current. As that warm water flows through our current, what that's going to do is cause our southern Indian Ocean to be warm relative to our northern Indian Ocean, weakening this energy gradient that was controlling our rain belt before, and allowing our rain belt to drift south. Right? And then our rain belt drifts south. Now it's going to spend a lot more time over Malawi and a lot less time over these northern sites. So Malawi is going to get wet and those northern sites are going to get dry. And at the same time, this current here that used to circles Antarctica, as that it's going to be pushed northward, causing much cooler waters to move across southern Africa. And those sites are going to get drier. So during our glacial, we have a situation where Malawi is wet, north and south is dry. And that's because we have a warmer southern Indian Ocean, where a rain belt has drifted south, and it spent more time over Malawi and less time over the north. And this current here has been pushing northward, causing cold water to flow off southern Africa, making those sites drier. So the conclusion for this part is that wet conditions are associated with cool sea surface temperatures and vice versa, producing an anti-phase relationship with sites to the north and south. To our knowledge, this anti-phase relationship was not captured by climate models, meaning that they could be missing key components of the system. And theories that link human development to climate need to account for this anti-phase relationship. Oftentimes, people who study human development in Eastern Africa, they'll talk about things like high lake phases and low lake phases. What we found is that those phases don't exist at all. There are no periods where all of East Africa is wet or all of East Africa is dry. Okay. So the next part of my talk here, I'll tell you a bit about our work on Lake Vesuvio. So Lake Vesuvio, the problem is much similar to Lake Malawi, where it's an old, long drill core, and we're refining the chronology, but it's a really very different system. So Lake Vesuvio is all the way in West Africa. We're moving to a whole new area. It's one of only a few natural lakes in Western Africa, and it was formed by a bolide impact in the big asteroid impact 1.07 million years ago. This lake is 80 meters deep and has a diameter of 8 kilometers. So our lake is relatively small and very deep for its size because it was formed by this impact event. This is really important for the sedimentation dynamics in our system. So our lake fills most of the 10-kilometer diameter impact structure. So it effectively has a very small catchment. But during dry intervals, when our lake is much lower, we're going to have a much larger catchment. And effectively have a lot more sediment entering our lake. Right? So we have a much smaller lake to deal with now. And how much sediment enters our lake should be highly linked to lake level. Right? So Lake Malawi, this core was collected back around 200 years ago. It's around 300 meters long. It's also among the oldest, most continuous sediment records from the continental tropics, but its use has been limited due to problems with the chronology. Like Lake Malawi, we've got robust dating in our upper parts of the core, And there have been several attempts at dating deeper portions, but none of them have been fully successful. So we're looking at a similar plot here. We have depth on our x-axis, age on our y-axis. And here's all the times people have tried to date deeper parts of the core. They've either linearly interpolated over large parts of the core or produced chronology by forcing cyclicity found in what they thought were well-dated parts of the core through the rest of the record. But there are two key problems with that method. One is that what they did was assume what was driving the system. They assumed they picked a driver of their hydroclimatic signal, and then assumed that their hydroclimatic signal must match that driver in order to generate a chronology. But if we then want to ask questions about what's driving our system, it becomes a circular argument, and you can't really do it. The other problem is that they were using some old ages that were actually miscalculated. And we can fix them now. Because they base all their assumptions on these bad ages, and then have to question everything. Right. So here's our working Lake Vesuvio age model. All of these red ones are new ages, and these blue ones are ages that we recalculated. Moving forward, so when this core was first collected, when Malawi and Vesuvio, they were collected around the same time. And the original goal was to be able to compare records from opposite ends of the continent. People could never do that because of the chronological uncertainty. But now that we're getting there, we've got there with Malawi, and we're getting there with Vesuvio, we can start to do that. We're looking at here is potassium titanium for those cores. That's just the upper part. And we can start to think about how these records on either side of the continent compare, whether they have the same driver or different drivers, etc. So moving forward with our lake on Lake Vesuvio, we're not done with the chronology. We're going to get some more luminescence data, more luminescence ages. We're going to try and date the deeper portion of the core using landscape evolution modeling. That's the part where I talked about how when our lake is very shallow, we're going to have lots of sediment going into our core. And when our lake when our lake is very high, we won't have very much sediment going in. What we're going to try and do is use understanding of that relationship once we have a good chronology for the upper half of the core to back out and figure out figure out the chronology for the bottom half of the core and eventual regional integration with other sites. Okay, so just the last bit here, I'm going to tell you just a little bit about. This is a very different project, but it's the one that I'm at Haifa to work on. This is on Lake Tanganyika and nutrient cycling. So we're moving all the way back to East Africa, and we're thinking about a very different time scale and a very different question now. Okay. So Lake Tanganyika, like Lake Malawi, it's gigantic, right? This one is 675 kilometers long and has a maximum depth of 1,500 meters. It crosses through 4 different countries, and it's a vital source of protein, fresh water, and economic development for the people living in this region. Right. So, unfortunately, however, over the past several decades, this lake that's really important for this region, right? Regions that don't necessarily have a lot of resources, right? We're talking about the Democratic Republic of the Congo, Burundi, Tanzania, and Zambia. Burundi and the DRC in particular, really rely on the fishery as a source of income and as a source of food. But over the past several decades, we've seen major reductions in the lake, most starkly seen in reductions in the fish catch rate. By some estimates, fish catches have reduced by half, and people really aren't sure why. The cause of the decline is debated. It might be changing fishing practices or climatic variability or changes in land use. You've seen increase in population. It might just be an overfishing issue. You've seen changes to the way people use the land. A lot of the land has been stripped for cassava farming, and it means you have a lot more sediment going into your lake that could hurt fish spawning habitats. Or we've got global warming, where now our lake is warmer, it's more stratified, and you have less nutrients moving around. And right now we're not sure which of those options it is. So what we're doing to address that problem is use proxy phosphorus cycling, carbon and nitrogen isotopes, lipid biomarkers, XRF, and fecal biomarkers from 2 sediment cores to try and ask the question if historic human activity on the landscape would have influenced nutrient cycling and thus the fishery in the past, And if not, what was driving the changes in the fishery? So what we're trying to do is understand how our fishery has changed over the last 2,000 years and what produced those changes. So for this project, we have two cores, both going back around 2,000 years. They were selected because they have sets of previously published proxies that we'll be able to use for comparison, and because they're at just the right points to help us record depths in the oxycline. And our data is still coming in. This is around 25% of what will be our phosphorus cycling data. So I'm just going to tell you a bit how that works. We're still working through the interpretations. But when we're talking about proxies of phosphorus cycling, there are different kinds of phosphorus bound in different ways. We have iron-bound phosphorus, autogenic, detrital, organic, and total phosphorus. And the relationships between these different kinds of phosphorus are driven and linked by productivity, organic matter degradation, and redox conditions. And by studying the changes in abundance and relative abundance of these different kinds of phosphorus, we can study these processes. And the other main proxy that we're going to have are lipid biomarkers. This is around 20 data points. We'll have around 120 once it's all said and done. And they're a set of biologically derived proxies that can inform on redox conditions, terrestrial input, and things like temperature and such. Moving forward on that project, we're going to complete those data sets. And our eventual goal is to get fecal biomarkers that could tell us about human presence in the landscape. Well, that's sort of an open question for whether we'll be able to get that data set or not. And that's everything. Time for questions. Yeah.
Thank you very much, Laura, and ask questions.
On the meteor impact, how is there that much sediment? There's no tectonic depression.
So the hole was caused by the meteor left a hole because we've had a lake there for the last million years.
Yeah, so why hasn't the lake filled up?
Well, it has
with the sediment.
It has. Eventually, it will fill totally.
Okay.
It's been getting that's another part of it is that the lake has been getting the basin has been getting smaller and smaller throughout its entire history. Eventually, there will be no lake anymore, but there's still lake there. It was a really deep hole and it's been getting shallow.
Yes, one time I think you're mentioning about how in East Africa, the earlier position was lake level rise and fall, and you mentioned there wasn't like a time where it rose and it fell like that. Like there wasn't a time where it completely disappeared. I mean, can I ask what methodology you used for that, or I mean?
So what I'm saying is that sometimes people will talk about high lake phases and low lake phases in East Africa and say all of the lakes were high now and all of the lakes were low. But we've seen that our lake behaves differently than the others. And it covers such a large expanse that it's a really big part of East Africa that we see is behaving in opposition to the others. So we know that there's these high lake phases and low lake phases. People will say sometimes, they'll try and link and say, like, humans developed more during dry intervals. We saw a lot more changes and, like, say, brain growth or changes in new technologies when it was dry and conditions were hard. But we're showing here that there weren't periods where it was hard everywhere. There were periods where it was hard in some places and good in others, but it wasn't like it was dry everywhere at the same time. That didn't happen.
Questions? Are you shy?
What I know, OSL like requires darkness, so if the things the inside of the core not insulated enough.
Yes, the way I collect these, our cores were split and they've been stored. All the outside of the sediment has been exposed to sunlight. What I do is stick a metal tube inside, and you tap each end, and then you take that into a dark room. And you cut off the outside, say 5 millimeters of the end that's exposed to sunlight, and the inside is all right. They've done studies, and because we're talking about like really fine-grained sediment, that fine-grained sediment is sort of like a dark wall, and it only takes about 1 millimeter of sediment to stop all the sunlight from penetrating. So we just cut out those outside sunlight portions and keep it out.
Are there questions from the audience on Zoom? And now I see the faces, so you can say no or yes in the face. No. No, I don't see. Okay, thank you. Okay, okie doke. So thank you, Laura. Again. And see you next week. I think next week we have another one. Yes. See you.